or very lopsided balance. In the liver, on the other hand, the oxaloacetate and the acetyl-CoA balance is way off, is skewed, because the liver is the only organ that can make glucose, not the other organs. Right? So in the other organs, the oxaloacetate is left alone. The oxaloacetate in the mitochondria is left alone. Okay? So whatever acetyl-CoA that's being fed into the mitochondria and the other organs, it will just react in the TCA cycle, no problem. Right? Now the problem is that acetyl-CoA is low in the other organs. In the liver, on the other hand, oxaloacetate is being taken out to make glucose in the process called gluconeogenesis. So this balance is completely skewed, whereby you have very low oxaloacetate and very, very high acetyl-CoA balance. Acetyl-CoA concentration is sky high because of the breakdown of fatty acids. Right? So you can't just let the acetyl-CoA sit there in the liver, right? It can be used constructively. So the liver changes it to a form which we call ketone bodies that can easily move out of the liver. Okay, so it can easily diffuse out of the liver and get transported to elsewhere in the body. Alright? Okay? So acetyl-CoA on its own, it is an activated compound, high energy. It probably will not make it to the other organs because it's going to probably react with all the other things along the way in the tissue and the blood. Okay, so you need to change it to something non-toxic, soluble, relatively non-reactive. Let it move. Let it go to its destination first before it's being used. Alright, so therefore, all the ketone, the liver will change acetyl-CoA to ketone bodies. Ketone bodies will move out of the liver to the skeletal muscles, to the brain, to wherever, and extrahepatic, anywhere out of the liver. And then this will change the acetyl-CoA and then feed into the TCA cycle. Okay? Alright, so this is really fuel economy for the body. Right? And it is extremely intelligent. Right? So we can still function. You have to really go through your pathways now to appreciate it. Alright? One of the things that you must ask yourself when you're revising is to ask yourself, where is this pathway occurring? Where in many levels? Okay? Where, which organ? Where, which cell in the organ? Alright? And then where, which organ now in the cell? Alright? So which compartment in the cell? So glycolysis we started, right? In all the cells. Glycolysis can occur in all the cells. And which compartment? Which compartment? Glycolysis. Which compartment? Cytosol or mitochondria? Cytosol, correct? Glycolysis is outside the mitochondria. Cytosol in the cytoplasm. And then we go into TCA cycle. Now, TCA cycle, which compartment is it? Mitochondria, correct. Now we go into mitochondria. Then we go into fatty acid mobilization. We say that fatty acid is also in the mitochondria. Correct? Fatty acid is also in the mitochondria. And then which organ? It can be in any organ. It can be in the myocyte. It can be in the hepatocyte. Right? Correct? Myocyte also have lipid droplets, right? But not much. Not much. Because myocyte is mainly for your muscle cells. Right? But in the liver, now, for the fatty acid, there's something special. In the liver now, you have a lot of lipid droplets deposited there. Alright? Okay, so especially now the liver is being involved, and that is where now with fatty acid metabolism, then you get this ketone body mobilization. Okay? And then now we go into amino acid. Okay, amino acid mobilization, the same thing, we ask ourselves, which compartment? Cytosol, mitochondria, where? Okay? Right? So, and then we ask ourselves, which organ? Right? So, for amino acid metabolism, the same thing. Also the liver. Most of the amino acids are broken down in the liver. So it's important, you must protect your liver. Right? Liver is a very important organ. Right? So, Yeah, by the way, liver, besides, there are only 2 organs in your body that can regenerate itself. Okay, one is your skin and the other is your liver. Alright, so if you see read Greek mythology, there will be, I can't remember this which Greek god it is or whatever, but there's this picture of a vulture pecking at the liver, right? And then the person doesn't die because the liver regenerates, even though the vulture was eating the liver. Right? Okay, so it is an organ that can regenerate. Therefore, we have liver transplant. Right? We have liver transplant. We put another person's liver into someone's liver which is not functioning and try to regenerate the liver. Okay? So for amino acid degradation, balanced diet, again, let's go back to balanced diet, carbohydrates, fats, and proteins. So the amino acids will be from your proteins, right? Okay? But the only difference, okay, carbohydrates can be stored, right, as glycogen. Fats can be stored, right, in your adipocytes. Proteins can be stored or not? Cannot. Okay? Problem. Right? Proteins, the amino acids cannot be stored. Right? So you have to eat it every day. Right? So amino acids cannot be stored. But so amino acids, when you ingest it, it will be broken down, right? If it will be, of course, utilized for biosynthesis, right? You will need to build up extra proteins in your body, muscles, etc. But if they are not required, right? If they are not needed for new protein synthesis, all the amino acids have to be removed from your body. Okay? And sometimes your cells, your proteins, your enzymes break down, gets degraded, right? Renew. It will be broken down and it will also give you amino acids, right? Or when you are really like a steak lover, you eat a lot of beef, steaks, and meat, okay? Rich in protein. When your diet is rich in protein, and ingested amino acids exceed the body's needs for protein synthesis, it will also be degraded and removed, right? Because it cannot be stored, not like fats and carbohydrates. Alright, okay? That's why Atkins diet asks you to just eat meat, right? Because it cannot be stored. Alright, okay? So Atkins diet says you can eat all sorts of meat, just don't eat carbohydrates. Alright, fats and meat is okay. So now, or during starvation, alright, during starvation or in uncontrolled diabetes, again, the carbohydrates is not available or not properly utilized. Amino acids will also be broken down. So these 3 scenarios. So we need to know how amino acids can be broken down. So intracellular protein or diet, protein, and then you have amino acids here. Okay, you know your 20 amino acids, yeah? Okay? So now the difference between glucose, fatty acids, and amino acids. Chemical structure. What's the difference? Now I'm trying to tell you how do you study laterally to compare these, on everything that you're learning. Glucose, fatty acids, and amino acids. What's so special about amino acids in the chemical structure? That is a concern for us in the breakdown. The amino group. Do you notice? Carbohydrates don't have amino group, right? You don't see them. It's all just C and H and O. Fatty acids also, just C, H, and O. No N. Amino acids is the first one with the N. Okay? Right? So now we have to figure out how to remove this amino group. Right? So in amino acid metabolism, this is our main concern. How to remove ammonia. Okay? Of course, ammonium ion, ammonia or amino group in the form of ammonium can be used to synthesize the amino acids. It can be used to synthesize the nucleotides, right? Nucleotides also has amino group, right? And biological amines. Okay? It will be used for biosynthesis. But if there's a lot of excess, then we need to remove it somehow. And remove it meaning out of the body. Okay? And out of the body, it is in the form of urea in the urine. Okay? Urea in the urine. So this ammonia we'll go through today. We'll look at urea cycle, right? How it gets removed from the body as urea out, okay? Out as urea. This is like completely out. Well, there is a lot of energy to be harvested from amino acids too. So the carbon atoms, the C and the H, we want it, okay? We don't want to waste anything. Right, the C and the H, we call it the carbon atoms, right? These carbon atoms will, the same thing, okay? Because the C and H will undergo complete oxidation to become carbon dioxide and water. And in the process, produce ATP. Again, it converges back into the citric acid cycle. Okay, it converges into your citric acid cycle. And then out as carbon dioxide and water, and then ATP. This is energy producing. Right, energy producing, this is a waste product. And besides that, we also have, out of this carbon atom, we can harvest 3 to 4 carbon units for gluconeogenesis to glucose. Oxaloacetate is one example, right? See, the oxaloacetate, this can be derived from the amino acids, right? From the amino acids that you eat too. We just talked about this, right? This is happening in the liver. We use this as an intermediate to make glucose when the body does not have enough glucose. Now, and then this two cycles, citric acid cycle and urea cycle, is connected together. Alright, it's connected together by a shunt called aspartate argininosuccinate shunt. Or in short, this is what I was mentioning. This is the Krebs bicycle. Alright? So this is Krebs cycle, and this with together the urea cycle, it makes a Krebs bicycle. See the wheels of the bicycle, right? It turns each other on and then move. Okay, move, like a pendulum bike. Okay, so this is the Krebs bicycle. So we'll look at how it's being connected. Okay, urea and citric acid.
How you laugh so loud?
So you see, everything converges back to citric acid. How important this cycle is, right? You have to understand. Okay?
So
you know the objective now, okay? Previously it was all about acetyl-CoA. But amino acids switch a little bit, okay? We have to be concerned with removing the ammonia, the amino group. Why? Because excess ammonia in the body is harmful. Okay? You cannot have very simple, you tell me, okay, why don't we just remove it as ammonia? After all, it's NH3, right? Yeah, but we can't. We can't let it accumulate in our body as ammonia itself. Okay, because it's very toxic. Again, so we need to disguise it. Okay, we need to convert it with all these biochemical processes to something that is soluble, that is not harmful, non-toxic, and then remove it in the urine. Okay, so that's the whole idea. Okay, now we look at the compartmentalization in the organs. Yeah, okay. So see, most amino acids is metabolized in the liver, like your fatty acid, beta oxidation. Okay, so over here, then this is the blood. Okay, this is blood, the vessels, and then your muscles. Right, we talk a lot about your muscles because we move a lot, right? You are not like sleeping like Big Brown Bear every day. Although some of you look like that now. Life in Korea. But you're always moving around, right? From lecture hall to lecture hall. So your muscles are very important, right? Respiratory muscles needs to contract, deflate, inflate. Okay, muscles are very important, right? So we always talk about muscles. Now, okay, so now here, let's start with maybe hepatocytes, right? Okay, because liver metabolize amino acids. You eat proteins, gets broken down the amino acids in your digestive system. It reaches your liver through the portal vein. Then in the hepatocytes, which is your liver cells, in the cytosol, the amino groups from most of the amino acids is transferred to alpha ketoglutarate. We've seen this before. Alpha ketoglutarate. Which cycle? TCA cycle. Okay, it's one of the intermediates in the TCA cycle. Alpha ketoglutarate. To form glutamate. Okay, look at here. Okay, so in the liver, okay, you have over here alpha ketoglutarate transaminated. Okay, the amino groups form. most of your amino acids is transferred to alpha ketoglutarate to become glutamate. Okay? Then this glutamate will enter the mitochondria and give up this amino group to form ammonium or ammonia. Okay, ammonium ions. Right? And it will also give rise through a transamination process to aspartate. These two combined will make urea. Then urea goes to urine. Okay, urea to urine. Simple, huh? Okay? So now, with this sentence, then you know for amino acid metabolism, we have both cytosol and mitochondria involved. Okay? Previously, it's either cytosol or mitochondria, but in this case, it's both cytosol and then to mitochondria. First in the cytosol, then to mitochondria. Okay? So in the muscles, okay, you have glucose, glycogen becoming pyruvate, etc. And then you have your amino acids, right? Okay? So in the muscles, excess amino groups is transferred to pyruvate. Okay, the amino acid is transferred to pyruvate to form alanine, and the alanine moves out of the muscle to the liver. Alright, and then alanine passes on the amino group to alpha ketoglutarate, which becomes glutamate, and then alanine becomes to pyruvate. So this whole process is like passing the parcel again, because right, I just mentioned we cannot remove it as ammonia gas, so we must form a cycle to pass on the amino group, okay, and let all these molecules carry the amino group in a temporary fashion, okay, in a temporary fashion so that they can move out to the liver and finally to become end product urea. Okay, we will look at this in detail, right? Okay. So in other tissues, so you have in the muscle, it is transported as alanine, okay, and in the liver, it is transported as glutamate. The amino group is transported as glutamate, and in the muscle, in other tissues, not these two, it is transported as glutamine. Okay, so alanine and glutamine will both go to the liver. Okay, wherever they are, they will all be channeled to the liver for this ureal cycle. Okay, so liver is like your garbage center, right? The final garbage center. Everything gets transported there to be removed. Okay, so this is what we'll look at it. Hopefully we get there. Ureal cycle. Okay, probably not, but anyway. So already 11:22. Okay, so now, did you see that when I was talking about passing the parcel? This parcel is amino group. So we are just transferring, okay, amino group from one molecule to another molecule. There's no loss of amino group. Transferring, it's a transferring process. Pass the parcel, like yeah. And over here, okay, all these reactions. This reaction is called transamination. Okay, we don't lose any amino group. We just transfer it from one molecule to another molecule, and they're catalyzed by enzymes called transaminases or aminotransferases. Right? So we have the amino group, alpha amino group, transferred to alpha carbon. Right? This alpha carbon is relative to the carbonyl group, CO group. We will look at it later. Remember this alpha. Like a fatty acid, alpha means the one right next to the carbonyl group. Alpha, then further away is beta. Okay? So alpha ketoglutarate, right? It is transferred to alpha ketoglutarate, leaving behind the keto acid. Okay? We will also look at what is the keto acid. Why do we do that? Okay, the general purpose is to collect all the amino groups from many different amino acids in the form of L-glutamate in the liver. Okay, this is the main objective, right? Because we said that the difference between amino acids with glucose and fatty acid is this troublesome amino group. So we collect them. Okay, we collect them in the form of glutamate, okay, in the liver, and then remove it. The glutamate is then function as an amino group donor, okay, for either biosynthetic pathways to make amino acids or nucleotides, any molecule that needs an amine group, and or to remove it in the urine. Okay? So this transamination process requires a coenzyme called pyridoxal phosphate, which is another vitamin. Okay, so you see that your vitamin B's are very important, right? So vitamin B's are very important. This is B6, right? Previously we have seen your coenzymes, cofactors, B5, B2, B3. Okay, now we have B6, pyridoxal phosphate. So it looks like that. Don't go crazy over memorizing structure. Okay, for amino acids, so fatty acid don't need to memorize structure, correct? You just need to know how to move your double bonds. Amino acids, all you require is what Dr. Ho require you to remember the 20 amino acid structure. Okay? All the intermediates, you don't really need it. I will give it to you if I ask a question on that. Okay? You will see, like your tutorial, I always give the structure and then let you compare to a drug molecule, for example. Okay? Because I think it's more important to understand, okay, than to memorize it. So over here, pyridoxine looks like this. Okay, and it goes through a whole cycle of conversion process to become first phosphate. Okay, so you look over here, the 5' carbon is here and becomes a phosphate, pyridoxal 5' phosphate. Right, and this is a cofactor that is associated with the aminotransferase enzyme. Right, we said that the process is related to aminotransferase. So the aminotransferase enzyme is here, and the cofactor will sit in the structure of the enzyme or gets connected to the enzyme via a lysine residue in the enzyme. Okay, so this is the lysine residue in the enzyme, and the cofactor gets bound to the lysine residue in the enzyme. And this moiety, chemical moiety, is called a shift base. So shift base looks like that, okay, CN. The shift base has this CN double bond over here, R1, R2, R3. Doesn't matter what R it is, okay, could be hydrogen, could be methyl. But the shift base looks like that, the CN characteristic. Okay, so you see a CN over here, R1, R2, R3 over here. Okay, R1, R2, R3. Okay, this is the shift base for C double bond. So the shift base will then be, see the shift base will then become this amine, okay? This would be the amino group that we want to carry. Okay, so we have to pass the parcel through the enzyme using this coenzyme. Okay? Pyridoxal phosphate. Let's see how it works, yeah? Okay, look at the scheme. Alright, first the pyridoxal phosphate enzyme is connected to the enzyme, okay, as a cofactor through the lysine residue. Then somehow the amino acid will pass on the amino group to this cofactor in this case. Let's see how it happens. Alright? Okay? Okay, take a deep breath, okay? So, again, objective, pass on the amino group. So this is your characteristic amino acid structure, right? COO minus. Why do we call it alpha amino acid? Because carbonyl group, with reference carbonyl group, the first carbon next to it is called the alpha carbon, right? So this is alpha amino acid, right? Alpha carbon is here. Okay, right, alpha. So over here, this is the amine group. This is our objective. Okay, this forms the carbon skeleton that we can use for biosynthetic process, TCA cycle, but this is the trouble, okay? We need to remove this, okay? Right? So now, the amino acid comes in and donates, yeah? You see this enzyme, pyridoxal phosphate, bound, the pyridoxal phosphate is bound to the enzyme through the lysine residue to form a shift base, okay? And then the amino acid comes in and gets attached. Okay? Gets attached here. See the amino group, the whole amino acid interacts with the paradoxal phosphate and form a shift base. Okay, this is the intermediate. Then it gets rearranged, okay, it gets rearranged with the end result of this alpha keto acid. Why do we call it alpha? So alpha keto acid. Alright, this becomes CO. Alpha keto group acid. Okay? Alpha keto group acid. It offloads, the amino acid offloads its amine group to the enzyme, to the paradoxal phosphate over here. Right? Offloaded. And then leaves as an alpha keto acid. Right? At the end, the amino group is now being passed on to the coenzyme now. Right? But the paradoxal phosphate cannot carry it all the time. It has to continue the process and bring it through to removal. Right? So then, over here, another substrate, which is now an alpha keto acid, then comes in again to regenerate. Okay, to regenerate this bond, right? And then it gets rearrangement, and the amine group gets passed on to the alpha keto acid. Okay, so do you see this musical chair kind of, or passing puzzle game? Right, so imagine this is your, let's say you have excess, you break down the protein, this is alanine. Excess alanine, you want to break down alanine, right? Okay, so alanine will leave as an alpha keto acid, passing on the amino acid. And in the liver, alpha keto glutarate will mop up, okay, will mop up the amino group, okay, to become glutamate. Okay, alpha keto acid will become glutamate by absorbing or by taking on the amino group. Alright? So in the end, after this whole process, the amino group from alanine becomes glutamate in the liver. Okay? Becomes glutamate. Alpha keto, can you put alpha keto glutarate over here? And then it becomes glutamate over here. And then this glutamate will then be sent into the urea cycle for removal. Okay, so this is the transamination process, right? And this whole process requires a shift base formation and rearrangement, okay? Rearrangement to become an alpha keto acid. Alpha keto acid. Okay. So, if this is an alanine, the enzyme will be called, this is the amino donor, amino group donor. So the enzyme will be called alanine aminotransferase. If this is a valine, then the enzyme will be called valine amino transferase. Okay, so the enzyme aminotransferase is always named after the donor, the amino acid donor. Okay, if it's an aspartate, then it will be aspartate aminotransferase. So I have another slide I gave in the notes. It's actually wrong. I wrote transamination and then there was a reaction scheme. Okay, that is actually deamination. So it will come out later. So just KIV first, alright? KIV first. Don't look at the slide. We have to move that slide later and changed. Okay. So over here, alright, so we ended with glutamate, right? Okay, so the amino groups from the amino acids are collected in the liver in the form of glutamate molecules. Okay, glutamate molecules. And the glutamate is then transported from the cytosol to the mitochondria, and now it undergoes oxidative deamination. Okay, with by L-glutamate dehydrogenase. Now, this is a deamination process. It's no longer a transamination process. What's the difference? Remember, I said transamination means passing the parcel. You don't lose any amino group. You transfer the amino group. Right? You transfer the amino group from one substrate to the product, and product to the substrate. No loss. But deamination over here means it's lost. Amino group is completely lost. Okay? It's lost from the substrate. Okay, so we'll look at how this is. Again, now you see NADP comes out. NADP, okay, appears again. right? So NADP was in fatty acid oxidation, right, as a reductive process. Now it comes back again in amino acid. So this is the oxidative deamination process. Okay, over here, now finally your glutamate structure. You probably already know. Okay, this is your R group. This is your amine group. This is the one that I made a mistake. I wrote transamination, but it's wrong. Can you change it to oxidative deamination? Did you see it somewhere down the slide? I think I made a mistake. I wrote transamination, but this is an oxidative deamination process. The title should be changed. Yeah, title should be changed. It's not transamination anymore. Okay? Yeah. And this is also wrong. It's glutamate dehydrogenase. Okay? Right. So over here, just now, the transamination process was using pyridoxal phosphate and an amino transferase, right? Okay. So, but over here, the glutamate dehydrogenase. Okay, this is your glutamate structure, the R group, right? NH2 amine group and the carboxylic acid group, and this is the R. Okay. The R here is CH2, COO minus. This is glutamate. Right? What's the difference between glutamate and alpha glutamate? Very similar. What's the difference? Okay, let's examine it now. The R group is the same. The difference is the alpha carbon. Glutamate is an amino group. Alpha ketoglutarate is a carbonyl group. Okay? You see? Same length. Okay, so if you remember one, you remember the other one. Okay, glutamate. Okay, over here, amino group. Alpha ketoglutarate is a carbonyl group. Okay, so this is called an alpha keto acid. Okay, over here, alpha keto acid. Easy to remember, right? This is R. Could be anything. R could be anything, but as long as you see the alpha carbon keto acid. So alpha ketoglutarate is the alpha keto acid. So the glutamate dehydrogenase will catalyze this reaction, right? Okay, through an amino group, CN amino group, okay, with the help of an equivalent, the NADP equivalent, right? Okay, right. So it loses its hydrogen. Okay, over here. And then it becomes the alpha keto acid and ammonium ion over here. Okay? So that's why this is not a transamination process. This is on its own now. Right? The transamination process, the amino group is always carried by some other molecule. But in the deamination process, this is removed. D means removed, right? The amino group is removed on its own as ammonium ion. Okay, so be very clear about process. Okay, the transamination process is for us to transport it, move it from other tissues to the liver. But once it reaches the liver in the form of glutamate, it will then be deaminated, okay, to remove as ammonium. Right? Okay. So the ammonium process, so ammonium is here, so we are here, glutamate, right? Glutamate dehydrogenase over here. Ammonium is here. Okay, right, we said already, this is quite troublesome. It will affect pH balance. It will affect it is toxic, right? It is toxic if you have too much of this. in the bloodstream. Therefore, it cannot be carried out like this. You cannot have ammonium ion just diffusing or moving into the kidneys and then remove as urine. Cannot. Remove in the urine, cannot. So you must figure a way out, okay, to disguise this molecule. So now, so this glutamate will become ammonia doesn't matter over here, okay? Now it will be in the mitochondria, right? It goes through the first part in the mitochondria, the urea cycle, goes through the first part to convert the ammonia into carbamoyl phosphate with the help of a bicarb, okay, and with the help of ATP. Where do we get the bicarb? Bicarbonate ion from. So the mitochondria produces your carbon dioxide, right, through respiration. So the carbon dioxide can dissolve into the fluids, into the liquid, and become bicarb ion. Okay, this is how it turns out. So the bicarb ion will combine with ammonia and then become carbamoyl phosphate with the help of ATP. Alright, okay. On the other hand, remember this alanine? Where did this alanine come from? The muscles, right? The alanine will go into the liver, offload the amino group to become glutamate, right? And then the alanine goes back to become pyruvate. Look at how similar the structure is again. Again, what is pyruvate? Alpha keto acid, right? Pyruvate is the alpha keto acid of alanine. See alanine over here? This is NH3. Right? Alanine is very simple, the structure. CH3, COO minus. The R group is a methyl group. Right? So the ammonia is here, and once you remove the ammonia, pass it on, it becomes an alpha keto acid. This is a transamination process. Okay? Transamination process. And the pyruvate goes back to the mitochondria. It can then be converted. It can either go through TCA cycle, or it could be converted to oxaloacetate. Oh, this is a TCA cycle, right? Or it could be converted directly using pyruvate carboxylase to become oxaloacetate, right? So oxaloacetate can become glucose, can also become amino acids. So oxaloacetate is quite versatile. In this case, this reaction scheme shows that oxaloacetate becomes aspartate, right? Not glucose anymore. Aspartate. We will see how important this process is. Alright? The process is quite long, we can't get to it today anymore. So we'll stop here. We'll stop here today. Alright? Okay? Hold the idea, yeah? Look at the amino acid. The ammonia group is here.
That time it was still manageable. This is really difficult.